Hello, I am Dr. Tanmay Deshpande. I am attached with Sir H.N. Reliance Foundation Hospital, Mumbai as a consultant medical geneticist. I welcome you today on this Facebook Live session for World DNA Day celebration. We are going to comprehensively talk about a genetic disorder and its basis and the tests to be chosen for a genetic disorder. A genetic disorder has to be identified for an individual to understand whether there is any prognostic factor, there is any recurrence risk, or there is any significant family history that has to be looked at. Let's go into the basics of genetics first. A cell has a nucleus which is considered as the central processing unit or the CPU of the cell. This cell inside contains the genetic material which in turn makes a protein which commands the cell to do a function. For example, any individual who has a disease called as beta thalassemia major, here the HBB gene fails to make a protein which in turn fails to produce new blood cells, and these individuals have to be transfused with blood every month, and hence a genetic disorder is considered very severe when the consequences are severe. A genetic disorder can be classified into a chromosomal cause or a single gene cause. Chromosomes are basic structures which have the genes inside them. So when we talk about a genetic disorder, there are two types of disorders that we grossly classify. One is a chromosomal cause and the second is a single gene cause. A chromosome is a structure which is present in every individual in pairs because one comes from the mother and one comes from the father. There are 22 pairs of autosomes and one pair of sex chromosomes. The sex chromosomes, if present in the form of two X's, makes the individual a biological female, and if it is present in the form of one X and one Y, it makes that individual a biological male. These chromosomes, when we unwind them, they have the single genes inside them. There are about 25,000 genes which are identified till date which form the functions of the body. These genes are also present in the form of pairs because again one copy will come from the mother and one copy will come from the father. When we talk about a chromosomal cause, it can be either a structural cause where one part of the chromosome goes and sits on the other part, or there can be a numerical problem. For example, a Down syndrome child has 3 pairs of chromosome instead of 2 on chromosome number 21. Now, when we talk about a chromosomal cause, it can be identified using tests like FISH, tests like karyotype, and tests like chromosomal microarray. When we talk about a single gene cause, the modes of inheritance determine the problems. For example, if both the pairs of a chromosome are affected, that individual is called to be affected with an autosomal recessive disorder. If one copy of a pair is affected, then this cause is called as an autosomal dominant type of inheritance, where only one copy, if affected, is enough to show a disease or its manifestations. There are similar disorders which are X-linked disorders, that is, the chromosome X has this gene which is affected, or Y-linked disorders where the chromosome number Y will have the affected genes. Now there are cases where we see that a random individual or sudden occurrence of a genetic problem in the family without having an individual family history. These kind of problems are called or known as de novo mutations. That is, a random occurrence or a new mutation which arrives in an individual randomly. To choose the right test, you need to take clinical manifestations, clinical symptoms, diagnostic tests, and a very significant family history into consideration. There are multiple tests which range from thousands of rupees to lakhs of rupees, for example, karyotype to whole genome sequencing. But which test has to be chosen for which disorder is curated by a clinical geneticist, and hence it is very important to meet a clinical geneticist or a genetic counselor to understand and curate the right test for you. I wish you all a very happy World DNA Day, and together I hope we can unwind the helix and help each other identifying genetic disorders and reducing the recurrence risk of these disorders. Thank you.